So now we are going to spend some time talking about staining specimens for microscopy and all the different types of microscopy. So here we are at chapter 2.4, staining microscopic specimens. So why do we stain specimens for microscopy? First of all, if we were to take cells and put them on a slide and then put them underneath the microscope, oftentimes, particularly when we're talking about light microscopy, but really any type of microscopy, there isn't enough contrast, so you're not actually going to be able to see much of anything because everything's kind of the same color. It's not like these big pictures that we see in textbooks when we look at a cell and we see the different parts, the nucleus and the Golgi apparatus and the mitochondria, and all the different parts are different colors, so it's nice and easy to see. That's not real life. In real life, everything is just kind of clearish because we're looking at something that's so small. So when we shine a light on it, it just kind of comes through clear and everything looks generally the same. There can be a little tiny bit of contrast just based on like the nucleus, for example, because there's a lot jam-packed into the nucleus, so it can be a little bit more like a shadowy area, but not really very clear. So what we need to do for bacteria or for other types of eukaryotic cells or prokaryotic cells, we need to provide contrast, and we provide contrast by utilizing different types of stains. And so there are different types of stains, different ways to stain, so we're going to go through those different things. First of all, before we actually get to the staining part, you have to prepare the specimen. So we have to prepare it for, and we're going to talk about light microscopy first, as the majority of the staining techniques that are used for light microscopy are very similar in other types, like fluorescence and the 2-photon and things like that. So when we are preparing specimens for light microscopy, so preparing specimens for light microscopy, there are two ways that we can do this. So there's the wet mount and then there's fixation. So a wet mount is pretty much as its name implies. You're going to take the specimen and it's placed on a slide in a drop of liquid. And then typically a cover slip will be applied, or it'll be a depression slide. And so you don't need a cover slip, or you can still put a cover slip on just to protect the microscope or the specimen. But a wet mount is wet, so it utilizes drops of liquid. So specimen is placed on a slide in a drop of liquid, and then use a cover slip. Now that doesn't mean that the drop of liquid is always something separate. So if, for example, we're looking at a blood slide, you might actually just take the drop of blood and then that's what's being looked at, or a urine slide. So take the drop of urine and put that on there. So other things though, however, we can take, if we take some sort of sample from the surface of a table or a cell phone or something like that to look at bacteria or yeast, then we would need to apply that drop of water and then, scrape the surface of something and then apply it to that liquid. So sometimes we add the liquid separately, sometimes the liquid comes with the sample. And there are also, so if we're talking about kind of something that we would be talking about in this class, we have our slide here, and then we can put our drop of liquid on there, and then we can use our loop that has some of our bacteria or whatever the sample is on it, and then we add that to the liquid. We kind of smear it around, smush it around, and then we can put a cover slip over it. Of course, we will be staining it prior to that too, but that's kind of the preparation would be the wet mount. So you put the drop of liquid in there, and then you're going to put the sample in there. Or there are other specialty slides that are called depression slides. So this would be like the typical slide here, but a depression slide has, as its name implies, a little depression in it, or an indent in it. And so then you can put the drop of liquid in here, and then the cover slip will just fit right over the surface. And a lot of times the depression slide is used for something like looking at something that's still living and moving around, like a paramecium or something, where it can then move around inside of that little depressed area, versus up here, once we put the liquid in here, we kind of smear it, then the cover slip covers it, and there's not a lot of space for it to move around at that point. Depending on the size, it can still move around, however. So we have the first one is a wet mount, and then the second one is fixing or fixation. In fixation, we are fixing the cells to the slide by using either heat or chemicals. So in this case, we are attaching the cells to the slide using heat or chemicals. So when we are utilizing heat fixation, this is often what is used when we're doing staining. In order to stain things appropriately, oftentimes, remember we already spoke in the microscope section about how when we stain something, we kill it. A wet mount, for example, like I mentioned with the paramecium here with the depression slide, in this case we might be utilizing something where we don't have to stain it, so we don't have to kill it. But if we're staining something, it's going to kill it. So we would utilize fixation at this point, because what we do is we apply the slide or the cells to the slide and then heat fix it or chemical fix it, and then that kills the slide, but it stops it. It stops it from going through any processes, and it just kind of puts everything on hold. And then since everything is on hold, then we can apply the stain and we see exactly where things were at that time. So a couple of things about fixation is that it kills the microorganism, but it retains the integrity of those cellular components. And this is because, so for example, heat fixing is what we typically use in our laboratory. And so when we do heat fixing, you're going to apply the sample to the slide and then just pass it over a flame just a couple of times. That just dries everything out, kind of seals it onto the slide, but it doesn't actually like burst the cell or start anything on fire. So when we heat fix it, as I was just mentioning, so when we heat fix it, you spread the smear. So you kind of spread a thin layer of the specimen. So spread a thin layer of the specimen on the slide. This is called a smear. And then you're going to heat it briefly. So again, pass it through a Bunsen burner flame just a couple of times. So, 2 to 3 times. Let's see, where were we? Spread a thin layer of specimen on the slide and heat briefly. So this is for heat fixation here. So then in the chemical fixation, we would add a specific type of chemical. So in chemical fixation, we would add a chemical like acetic acid or ethanol, formaldehyde, something like that. Then what this does is it still stops everything. It stops all the biochemical reactions, stabilizes those cell structures. But the chemical is typically used for tissues. So like skin cells, for example, we want to use something like formaldehyde to kind of stop everything versus heating things up, because sometimes that can destroy the cells. But chemically fixing can also denature some proteins as well. So we have to be really careful and choose wisely when we are utilizing these types of things. So for chemical, we add a chemical. So something like acetic acid or ethanol. And then we add the chemical to it, and then it's going to be prepared. So this also is going to, it stops the biochemical reactions. So stops biochemical reactions. So it stops the biochemical reactions. It can stabilize the cell structures. But it can denature those proteins. So it can kind of destroy some cellular structures. And then also remember that this is preferred for the tissue specimens. So preferred for tissues, rather than something like bacteria or archaea or yeast or things like that. So when we're talking about larger tissues, so like doing a liver biopsy or something like that, this would be a situation where we might use the chemical fixation versus heat fixation. Good. All right, so these are the steps here. This is the information that you should know regarding preparing the specimens for light microscopy. So now we're going to get into talking about the different types of stains and the different methods of staining. So staining, as we've been discussing it, what is staining? So staining is the process of coloring certain features of a specimen before examination under a light microscope. So we're going to, so it's the process of coloring certain features, and we're going to talk about how we can use these to do specific types of features rather than the entire cell. So certain features of a specimen before examination under the light microscope. So then our stains or our dyes are made up of certain types of salts. So a stain or a dye are made up of salts. And then there are typically two different types of salts in a particular stain or a dye. There's the ion that's going to color something, and then there's the uncolored ion. So the uncolored ion is called the counter ion, and then the colored ion is the chromophore. And we already talked a little bit about, or spoke a little bit about chromophores because we were talking about fluorochromophores when we were talking about fluorescence microscopy, so therefore we use dyes that have fluorochromophores. So the chromophore is the one that's going to be doing the coloring, that's why it's the colored ion, and then the one that is uncolored is the counter ion. So if the chromophore is positive, if it's a positively charged chromophore, so these are ions, right? Ions. So if it's a positively charged ion, positively charged chromophore, then we would call that a basic dye. And then it would, of course, have a counter ion, maybe something like hydroxide. If the chromophore is a negative ion, then that would be an acidic dye. And then, of course, we would have our counter ion. So, for example, it might be hydrogen or something else like that. So we have our basic dyes and we have our acidic dyes. And these are important, knowing whether or not the chromophore is a positive ion or a negative ion, because our positives and our negatives are going to interact differently with different parts of cells. So we would want to understand what it is that we're looking at so that we can understand if we need to use a basic dye or if we need to use an acidic dye, just depending on what it is that we're trying to look at, what it is that we're trying to create contrast for. So not only do we have to take into consideration whether or not the thing that we are trying to create contrast for is positive or negative, and therefore whether or not we want to use a positive or negative dye, we also need to understand that there are positive stains and negative stains. So a positive stain does not mean that we're using a positive dye or a basic dye. What it means is that in a positive stain, the dye is going to be absorbed by the object that we're looking at. So in a positive stain, what we're using is a dye that will be absorbed by the object of interest. So this is what's going to be absorbed by the object of interest. So if we're looking at bacteria and then we create a positive stain, or we decide to do a positive stain, what it's doing is it's staining the bacteria because it's a positive stain. We're staining the object of interest. On the other hand, we could use a negative stain. And a negative stain then would be the opposite of that. So this is a dye that is going to stain the background. So a dye that adds color to everything but the object of interest. Okay. So if we think about what we've just talked about here, so our colored ion or chromophore can either be positive or negative in charge, and then that gives us a basic or acidic dye. And then we can decide whether or not we want to stain the object of interest, meaning that we want to do a positive stain, or we can decide whether or not we want to do a negative stain, so we dye everything but our object of interest. We need to think about what the charges are and whether or not we want to use a specific dye. So for example, cell walls. So if we're talking about organisms, microorganisms like bacteria, for example, cell walls are usually negatively charged. So if cell walls are negatively charged, then we can use basic dyes. We can use basic dyes as positive stains. And if we wanted to do a negative stain, then we might want to use an acidic dye, right? So if we have our negatively charged cell wall, we have our basic dyes. Remember, basic dyes up here are positively charged colored ions, our chromophore. So then our negative and our positive are going to be attracted to each other. That means that this positively charged colored ion, chromophore, is going to attach to the cell walls of that bacteria or whatever it is that we're looking at. That's a cell that has a cell wall. And so that would be a positive stain. Now, if we wanted to then do a negative stain where we stain everything else, then we could use our acidic dyes, and then they're going to be opposed to each other, right? They're going to pull away from each other, and so we're not going to stain the object of interest, that cell wall. Instead, we're going to stain everything except for the cell wall. Good. So when we're talking about this, kind of as we've been talking about it now, we're talking about what's called simple staining. And with simple staining, we're using one single dye to stain. And usually it stains all the organisms in the sample, or it's staining the entire background. So in simple staining, it uses one dye to stain, and usually stains all organisms in the sample. So one dye, like we were just kind of mentioning. So we would use like we said up here, a basic dye to make our positive stain. We're just staining everything on there. All the cell walls are going to be stained the same color. That's a simple stain. Or the opposite. We use a negative stain, stain the entire background. So that's simple staining. You just apply one single dye to it. Now, on the other hand, we could have differential staining. In differential staining, in this case, we're using two or more dyes. And so in this case, what we can do is we can differentiate different organisms, so distinguish them from each other. So this uses two or more dyes to distinguish or differentiate organisms from each other. So in this case, there are some examples that you should be familiar with. And you should know there's in your text, there is a table that goes through all of the different types of dyes. So it goes through the simple dyes and the different types of simple dyes that there are, the simple staining techniques, and then there are the differential staining techniques, and you should know different types of chemicals that work in those ways. So for example, if we're talking about a simple staining, we could be talking about something like using crystal violet. We could be talking about malachite green. We could be talking about methylene blue, safranin, basic fuchsin, basic. And those are some of the examples that we could be used. Those are some of our basic dyes, for example. Because we were talking about basic for the cell walls there. So those are some examples of basic dyes that can be used as simple stains, but then some of those are also used in differential staining. But we have our acidic dyes. So let's say up here, I'm kind of running out of space. So let's put, these were basic, examples of basic dyes. Now let me put some examples down here of some acidic dyes. So we have our acidic fuchsin, eosin, and rose Bengal. Sorry, my notes are kind of all over here. So these are acidic dyes. So these are some examples of acidic dyes. These are some examples of basic dyes. And again, this is in the table in your text, so you should be familiar with those within the table as well. So getting back to our differential staining techniques, you should be familiar with these different staining techniques. The one you should be most familiar with is gram staining, and we'll talk a lot more about gram staining, not only in this chapter, but future chapters. So gram staining, also acid-fast staining, endospore, flagella, and capsule. So these are all different types of differential staining techniques. And what that means, again, is that we can distinguish between different organisms or different types or different parts of an organism. So in gram staining, we're going to differentiate between gram-positive and gram-negative organisms. In acid-fast, we're going to determine between acid-fast organisms and non-acid-fast organisms. In endospore staining, we're going to be able to stain the endospores. So it'll be endospores, and we'll be able to distinguish that with vegetative cells. Vegetative cells are those that are not producing endospores, that are just healthy, regular, everyday cells that are living their life being metabolically active, so vegetative cells. Flagella staining, of course, is going to stain a flagella or stain the flagella or flagellum, which is singular, and then that will stain it separate from the remainder of the cell. Capsule stain is going to allow us to see the capsule. So we can do capsule stains in two ways, and we'll talk about this in just a moment, but we can do it just a simple negative stain, and then we'll see the individual kind of clear cells that contain capsules, or we can do a stain where we can stain both the individual cell, so we can stain the cell, and a negative stain in the background. So then what we end up with is kind of like a halo effect. We stain the cell here, and then we can stain everything else in the background, and then we end up with the capsule, which is right around the cell. So it ends up kind of like a halo, versus over here in the negative stain, what we end up doing is just having the dye around here. All right, so let's get into some more details with these differential staining techniques. So the first one we are going to go over is the gram stain. And you should know the gram stain in detail. This is a very important one. It is kind of the first stop when trying to identify organisms, is utilizing a gram stain so that you can determine whether or not an organism is gram positive or if it's gram negative. And we are going to talk more about what it means to be gram positive, what it means to be gram negative. But at this point, you should understand that gram negative and gram positive means that there's a difference in the cell walls. So in gram stain, we're going to be distinguishing between different organisms that have different cell wall types. So this is a differential staining process to distinguish bacteria with different cell wall types. So we're going to distinguish between gram positive and gram negative. So in general, at this moment, what you should understand is that gram positive cells are going to be stained purple at the end here. So they're going to end up looking purple. And this is because they have a very thick layer of peptidoglycan in their cell wall. So remember that bacteria have peptidoglycan in their cell walls, And the difference between gram-positive bacteria and gram-negative bacteria is the amount of peptidoglycan. So in gram-positive bacteria, there's a very, very thick layer of peptidoglycan, and so it maintains a very dark purple stain. Our gram-negative cells end up looking pink, and that's because they have a thin layer of peptidoglycan. And so they do not maintain the purple color, and we're going to take a look at why that is in just now. So here are the steps, and you should be familiar with the steps and all of the different things that are associated with doing a gram stain. So the very first step is that we are going to add crystal violet. So, actually prior to the first step, remember we have to do the preparation. So when we do a preparation for a gram stain, I'm going to kind of put this in parentheses here. Let me get rid of this stuff. So first you want to prepare the slide. When you prepare the slide, what you want to do is you want to add your bacteria to the slide. Typically the way that's done is either coming from a broth, you put just a drop of broth so that you add the solution with the bacteria. If you're taking it as a sample, like scraping it from an agar plate, then you're going to add that to a drop of water, and we'll go into that in more detail in the laboratory portion. However, you want to prepare the slide, and then for gram stain, we are going to heat fix the slide. So remember, we already spoke about how we want to prepare, and we're going to have a heat fixed slide. So again, you add the sample, you pass it through the Bunsen burner flame a couple times, 2 to 3 times, and it just fixes it to the slide. Make sure that those cells stay still. All right, so then step 1 of the gram stain is we are going to add our primary stain. So add the primary stain. And the primary stain for gram staining is crystal violet. So you will add crystal violet, which is the primary stain. And then what you're going to do is you're going to let that sit for a period of time. We'll talk about those details later. You're going to apply the primary stain to that heat fixed slide, and then that's going to stain all cells purple. Then the second step is that you are going to add grams iodine. And grams iodine is what is called a mordant. And a mordant is something that's going to use to stabilize or set the stains in there. So we added the crystal violet. We kind of do a quick clean after the crystal violet, spray it down with water. Then we add the grams iodine, which is a mordant. So it's going to stabilize the dye within the cells. So it acts as a stabilizer, or to set, sets or stabilizes the dye. So we add our mordant, which is the grams iodine. It kind of traps the crystal violet in. Then what we're going to do is add decolorizer. Now when we add decolorizer, this is usually ethanol, or it can be a mixture of acetone and ethanol. And when we apply that solution, the cells that have their very, very thick peptidoglycan walls are not going to be affected. So the decolorizer is not going to decolorize the walls, the cell walls that have really thick layers of peptidoglycan. They maintain that crystal violet. However, the decolorizer is going to wash away the dye in the very thin layer of peptidoglycan. So our cells with thick peptidoglycan retain the dye. It's not washed away. Cells with thin peptidoglycan, the dye washes away. So even though we added the grams iodine, which is the mordant, there's just not enough of that dye in there, and it washes away with the decolorizer. Then the next step is when we are going to add our secondary stain. So our secondary stain, which is our counter stain. And the counter stain in this case is safranin. So when we apply the safranin, which is the counterstain, then what's going to happen is all of those decolorized cells, the cells that lost that crystal violet, are going to be stained with the safranin. And so they will look pink. So all of the decolorized cells pick up the stain. So all of the decolorized cells are going to pick up stain and turn pink, because the safranin is a kind of a deep red color, but what stays behind is the pink color. Now the colored cells from previously, so those that were not decolorized, those do pick up some of the pink, but you can't tell because there's so much purple. So they still have that purple from the crystal violet, even though some safranin may attach to it. It's not really noticeable, so they stay that really dark purple. So then what we end up with, again, here is we end up with pink and purple, and then remember that our purple is in the thick layer of peptidoglycan. So thick layer of peptidoglycan is purple, that's called gram-positive. And our thin layer of peptidoglycan is pink, and pink is our thin layer of peptidoglycan, which is gram-negative. So there are a couple of things to keep in mind when doing a gram stain. First, if the cells are old. So how about keep in mind, so one, old cells. So if you are using old cells, they may have damage to their cell walls. And so if they have damage to their cell walls, they may not have that big thick layer of peptidoglycan. So then they may end up all appearing gram-negative when they really do have some gram-positive. So damaged cell walls. So then they may end up appearing gram-negative when they're actually gram-positive. So you always want to make sure that you have a fresh culture of cells. And that's going to give you the best result, is a nice, healthy, fresh, growing culture of cells rather than some old cells. And the second thing to keep in mind, and this is a really important thing to keep in mind, particularly if you're taking the lab at our campus, is that decolorizing often happens. So if you use too much decolorizer, if you over decolorize, then that can be an error when you're doing a gram stain. And then what ends up is some of the organisms may appear gram negative, but they're actually gram positive. So something like too much decolorizer. So then they may appear gram negative, but they're actually gram positive. So if you can imagine, if you add lots and lots and lots of decolorizer and keep washing and washing and washing that away, even if you have that really big thick layer of peptidoglycan, you can wash so much of it away that you might actually start to see that red from the safranin or the pink from the safranin, and you may think that's gram negative, but in fact it's actually gram positive. So if you're identifying one single species, not doing two different species, if you're identifying one and you see both gram positive and gram negative cells, you may have added too much decolorizer, and it may actually just be a gram positive organism rather than a gram negative organism. All right, so that is our gram stain. So now we're going to move to our acid-fast stain. So our acid-fast stain is going to differentiate between a gram-positive cell that has this waxy mycolic acid in the cell walls and gram-positive cells that don't have the mycolic acid. So differentiate, because remember this is a differential stain. So differentiate between gram-positives with mycolic acid. Mycolic acid in the cell wall versus gram-positive without the mycolic acid. In the cell wall. So mycolic acid, we're going to get into more details about the structure of cells in Chapter 3. However, some cells have mycolic acid on the cell surface, so it is outside of the peptidoglycan. There's an additional layer of acid. So oftentimes what happens is if you are taking an organism and trying to identify it and you go through and you do a gram stain, sometimes you can end up getting kind of questionable results. It'll end up looking maybe like a mixture of gram positive and gram negative. And so then you can go in and do do an acid fast stain to see if the reason why you weren't getting a really good result, whether it's gram positive or gram negative, is because there's this additional mycolic acid layer on the outside of the cell that is kind of changing and messing with the gram stain results, because the mycolic acid actually is going to repel some of those stains. So you don't get a really good solid gram positive from it, even though it does have a very thick peptidoglycan wall. So in this case, what you can do is you can utilize an acid-fast stain in order to determine whether or not that mycolic acid is present. And if it is, then you know that you have a cell that has mycolic acid surrounding the cell wall. So there are two different methods of acid-fast staining. So there's the Ziel-Nielsen and the Kinyoun methods. So Ziel-Nielsen technique and the Kinyoun technique. So they're both acid-fast staining techniques. They both use carbofuscin as the primary stain, but one uses heat to kind of push in that carbofuscin in the acid-fast cells, which is the Ziel-Nielsen technique. So this uses heat. And then in the Kinyoun technique, it does not use heat. But they both use carbofuscin. So the step number one is to add or apply the carbofuscin. So the carbofuscin is actually a pink color. And in both cases, you're going to apply carbofuscin. In the Ziel-Nielsen technique, you actually do that in a heat setting. So typically there's a beaker on a hot plate and you have steam coming up from the beaker. You apply the carbofuscin and then you let that carbofuscin sit there as it's heating up, and it's kind of using that heat to push it further into the cells. The second step, then we're going to use a decolorizer. So the decolorizing agent is usually an acid or alcohol solution, so an acid alcohol solution. So we're going to wash it a little bit there. Then after we wash it, so that will decolorize the non-acid fast. So if we have a mixture of cells and we want to determine if there are some acid fast and there are not some acid fast, we'll add the carbofuscin, which is pink, and then that pink is going to be in the acid fast cells. So the acid fast cells are going to maintain that carbofuscin. Then we add the decolorizer, and any cell that does not have that waxy mycolic acid surface, they're going to decolorize, so they'll lose that pink color. And then we add our secondary stain, which is the counter stain. So our carbofuscin was our primary stain. So our secondary stain is our counter stain, and that's usually methylene blue. And so then the methylene blue is going to stain all of those that were decolorized, which means those are our non-acid fast organisms. So non-acid fast are going to be blue. So then what we end up with are, if we are looking at a mixture of bacteria, those that are acid fast, that have that mycolic acid in their outer layer cell wall, they are going to be pink. And then any other bacteria that's on there that's not acid fast is going to end up looking blue. So we can determine between those different types. All right, so our next type of stain is our capsule stain. So our capsule stain then is going to identify whether the organism has a capsule. Whoops. Identify a capsule. A capsule can be found in certain bacteria and certain yeasts. A capsule is something that is around the bacteria. It helps it to be more virulent. It helps it to attach to certain things, helps it to prevent dehydration. So it ends up making it more able to cause disease because it can hang out longer without dehydrating. It can attach itself to stuff more easily, and it can be passed along to other people more easily. So in our capsule stain, it can stain both bacteria, where we see capsules, and yeasts. Some yeasts have bacteria, or some yeasts have capsules, some bacteria have capsules. So it makes them more virulent. And our capsules, if we try to apply basic dyes to the capsule, it does not absorb the basic dyes. So what we typically see with a capsule stain is a negative stain used. So we dye the background, and then we see kind of these white blobs that are the capsules that are around our bacteria. So for the capsule stain, it's a negative stain. So we dye the background. So we are dyeing the background, and then this doesn't penetrate the capsules, so doesn't penetrate capsules. And so then what we end up seeing are these kind of halos around the cell. So we see halos around the cell. Now if we just do a primary stain, or a simple stain rather, if we do a simple stain, oftentimes if we do a simple stain, we'll just use nigrosin, or we can use India ink. So India ink or nigrosin, which gives us a nice dark black background. Sometimes then we can use India ink or nigrosin in a differential type of way where we can combine it. So where we can use, we can combine, we can do a positive and a negative stain. So what we can do in the positive stain is these would then color the cell, but not the capsule. And then in the negative stain, in this case, we would color the background. And so neither of these are going to be coloring the capsule, and then this is where we end up with that kind of halo. So if we were doing the capsule staining, we might see the bacteria itself being stained, and then we would have our black background of our negative stain looking something like this. So the negative stain doesn't go right up to the cell because it has that capsule around it. So it ends up looking something like that, like a halo around it, as I mentioned previously. All right, so the next stain is our endospore stain. And our endospore stain is going to allow us to identify endospores in bacteria. So identify endospores in bacteria. And endospores are structures that allow them to survive in harsh conditions. So structures them to survive in harsh conditions. And we'll talk a lot more about all of these things, the different types of capsules, the endospores, vegetative cells, all of these different things as we move through the future chapters. So, but right now you should understand that an endospore stain is going to identify endospores. Endospores are structures that cells are going to create. If they find themselves in kind of an iffy situation where there's not a good amount of nutrition or moisture or whatever it is, it can start to go through the process of making an endospore. And an endospore is going to allow them to live through a non-ideal situation. So if we try to use gram staining is not going to allow us to see endospores because it cannot penetrate through the endospore itself. So the endospore actually just looks like a clear area. So if we had a whole bunch of endospores on a slide, and we tried to stain it using a gram stain, then we would end up not seeing anything at all because the endospores don't pick up the dye. So we can say that gram stain alone cannot visualize endospores. They look clear. So instead, we have to do an endospore stain. And when we do the endospore stain, we do what's called a Schaefer-Fulton method. So Schaefer-Fulton method. So the Schaefer-Fulton method is one, we're going to add our primary stain. And our primary stain is our malachite green. And this malachite green we're going to use with heat. So similar to our acid-fast stain, we're going to let it sit on a beaker. We're going to allow that heat to then push that green color into the endospores. So it pushes that green color into the endospores. Then we are going to decolorize. So when we decolorize, then we can just use water to decolorize. And what this is going to do, it's going to remove the green from the cells, any vegetative cells, vegetative meaning just healthy cells that are living, that are not endospores, but it's not going to remove it from the endospore. So it removes the green from cells, but not endospores. Then lastly, we add our counter stain. So this is our secondary stain. And when we add our counter stain, this is usually safranin. And so then this ends up being a pink color. So all of the cells that are not endospores, our vegetative cells, are going to end up being pink. So we can see all of the different cells, and then we can see the actual endospores. A lot of times the endospores are within the vegetative cells, so you get to see them kind of in the process of making the endospores, and then you can see free endospores, and you can see free vegetative cells throughout there. So endospore stains are really important because we see these in different types of species, so we can see them or in different types of genera. Like Bacillus and Clostridium. And some of these Bacillus and Clostridium bacteria are very, very dangerous. So this is one technique that can be used after doing a gram stain, for example. Then one can go ahead and look and see if the bacterium is creating an endospore. And if it is, we can identify it as perhaps a Bacillus or a Clostridium. So some of the most common ones that do create endospores are Bacillus anthracis, which is anthrax. And our Clostridium, you may have heard of Clostridium difficile, or C. diff, as it's shortened to, which is very, very dangerous infection that some people get it. Typically, it's nosocomial, which means you get it from the hospital. It can be life-threatening. So the endospore stain can be very, very helpful in identifying both of those types of species or genera. All right, the next one is our Flagella staining. So again, this is going to be able to identify our flagella. Flagella is our tail-like cell structure. So tail-like cell structure used for motion. And we can see some bacteria have flagella, which is plural, or a flagellum, which is singular. So that would be something else that could be used to identify. So typically they're usually very, very thin, and there may only be one flagellum. If there are multiple, sometimes it can look a little hairy. But usually they're thin. Usually they're so thin that they cannot be seen without staining. So what we do for flagella staining, the first thing that we do is actually add a mordant. And so in this case, the mordant, typically tannic acid or potassium alum. In this case, it's going to coat the flagellum or flagella, and then it's going to thicken that because, again, it's very, very thin. So our mordant is going to coat and thicken the flagella or flagellum. So we do that first. Then our second step and last step is just our primary stain and our only stain. And in this case, we can use basic fuchsin or pyrrorosaniline. So then we can add either of those, and then we can identify and we can see the flagella. So it typically looks kind of pinkish, pink in color, and you can see all the little tails if they have them on there. So all the little flagella. All right, so then all of those different types of stains that we just went through are used for light microscopy. So something that we would use in our bright field microscope that you would typically see in a laboratory in a classroom, oftentimes used even in pathology as a light microscope or bright field microscope. But when we talk about other different types of microscopes that we have talked about, we do have to take into consideration the different ways that we have to prepare specimens for those microscopes. So for our electron microscopes, remember we had two different types of electron microscopes. Do you remember what they are? So we had TEM, which are transmission electron microscopes, TEM. Remember when we took a look at samples with TEM or under a TEM microscope, what we saw was very, very highly detailed three-dimensional structures, but internal structures because we had sliced them very thin. So when we are preparing them for TEM, the first thing we do is we embed the cells or embed the organism, so I'll just say cells, in a resin. We have to slice these so very thin that even using the thinnest, most delicate cutting tool, a diamond knife, would destroy the cells in order to use them for a TEM. So we have to embed them in resin, and then we have to dehydrate the sample. But you can't just take all the water out, otherwise you're going to ruin the way that the sample looks. So we dehydrate with ethanol soaks. So what we do is we increase the amount of ethanol every time we wash it and every time we soak it. And then what that does is the water is going to be displaced. We're going to replace the water and turn it into the ethanol. Water is going to turn into ethanol, and then that ethanol is going to be displaced by the resin. So it enters the cell and then it solidifies. And then it makes it much easier to cut. So then in step 3, then thin slices are cut with what's called a microtome or an ultra microtome. Which is a very precise cutting tool. An ultra microtome. So then, once we cut those very thin slices with the ultra microtome, then the samples are going to be fixed to copper or carbon grids. So the samples fixed to copper or carbon grids. And then lastly, we are going to stain them with electron-dense atoms. So we put those electron-dense atoms on there. Some examples the text gives are uranyl acetate or osmium tetroxide. So they have those electrons on there so that when we use our electron microscope, we can end up seeing all those fine details. So then the second type of electron microscope, remember that we had, is SEM or scanning electron microscope. So for our scanning electron microscope, remember this is three-dimensional images, very highly detailed, but it's on the surface. So for the scanning electron microscope, we also want to dehydrate the sample. So dehydrate with soaks of ethanol. In this case, we are going to dry it completely. So we're going to dry it completely to the critical point. With carbon dioxide in this case. So we're going to dehydrate it rather than with ethanol. Or after the ethanol, we're going to then dry it completely to the critical point with carbon dioxide. And then this is going to displace all the water. So then what we do, third step, is we sputter coat it. As we've already mentioned, we sputter coat it with metal atoms so that the specimen doesn't become charged by the SEM beam. So charged by SEM beam. All right, so then lastly, just kind of generally, we're going to say preparation for other microscopes. So all of the other microscopes that we had mentioned in the previous section, like fluorescence, for example, fluorescence, confocal, these are prepared similar to light microscopes. So similar to light microscope stains. But instead of using the specific dyes, instead the stains are fluorochromes. So remember, we already spoke about that a little bit. We have our fluorochromes, and then they're going to fluoresce when we're taking a look at this. So remember when we talked about, so our examples are immunofluorescence. So we had our dyes attached to the antibodies. And then when our dyes are attached to the antibodies, then they can fluoresce. So we can stain specific proteins, which these are proteins. We can stain the proteins, or we can stain DNA. And the example that your text gives is fish. So we can stain DNA, we can stain proteins, so like antibodies, for example. We can stain DNA with a fluorochrome. So it's basically we take a fluorochrome and we tag the DNA. And then in this case, we have what's called FISH, which is fluorescence in situ hybridization. In situ hybridization. That's where we get our FISH from, FISH. In this case, what we're doing is we're hybridizing with our DNA. And we are utilizing DNA that is tagged with fluorochromes, so we can see them in our fluorescence or confocal microscopes. We also had our 2-photon microscopy. In this case, it's similar to our fluorescence preparation, but we use IR dyes. So we use our infrared dyes to do our 2-photon microscopy. And then lastly, we have our STM. Remember, these are scanning probing. So these samples need to be on atomically clean and smooth surfaces. So remember, atomically, so at the atomic level. Then they are often coated with gold. And then we have a toluene vapor is used as a fixative. All right, and then we can utilize the skinning probing. All right, so this kind of wraps up all of our different types of staining mechanisms or staining procedures, methods, processes. So you can just know generally these other microscopes we just spoke about, but you should know in detail all of these different things that we spoke about in detail. So you should understand how to prepare a specimen for light microscopy, so the wet mount and fixation, and the two types of fixation, the heat and chemical. You should know the different types, what staining is, what a stain or a dye is composed of, so the salts, the different ions, the counterion, the chromophore, the differences between a basic and acidic dye, the difference between the positive stain, negative stain, why you would use different things like that. Understand the difference between a simple stain and a differential stain. You should know examples of basic dyes and acidic dyes. You should know gram stain very well, what it's going to provide us with as far as information is concerned, as well as what we would actually see, which is the purple and the pink, and what those mean. You should also know the steps, what's involved in it, and what to keep in mind. You should know the steps and understand why we would use an acid-fast stain, also a capsule stain, an endospores stain, and the Flagella stain. So you should know those different steps, why we would use those things. And then you should know a little bit of detail about the electron microscope stains, these different steps that we spoke about here, and then just that little bit mentioned about the other microscope stains and preparations.